populations within a species selected and adapted using the way. He also implemented novel statistical methods to get a better understanding of the genetics. In addition to these, he worked on transcriptomics, epigenomics, pharmacogenomics, and microbiomics, and a bit on human cancer. He has about 15 years of experience working in various aspects of genomics and bioinformatics. Dr. Sujan taught courses like genomics, genomic techniques and genetics, and also given hands-on training in structure and function of DNA, Mendelian genetics, PCR, gel electrophoresis, sequencing, polymorphism detection, linkage mapping, RT-PCR, RNA sequence analysis, in silico prediction and annotation of genes, forward and reverse genetics, transcriptomics, and also taught computer program languages also. Dr. Sujan co-supervised 17 PhD students and 4 master students. He organized more than 10 workshops in the department and also online to train personnel on association mapping and QTL mapping from North Dakota State University, USA. Dr. Sujan published his research findings in 45 international journals with very high impact factor journals like Nature Biotechnology, Nature Genetics. And Dr. Sujan is also academic editor for prestigious journals like PLOS ONE, a peer-reviewed open access scientific journal with impact factor of 3.24. Additionally, he authored 1 book chapter, 8 meeting proceedings, and also 46 presentations in international conferences. The publications of Dr. Sujan crossed, around 2,100 citations, and index of 21. We are very much fortunate to have you, sir, Dr. Sujan, on this online workshop on molecular data analysis through bioinformatic tools. I request Dr. Sujan Mamidi to start the highly informative session of the workshop, that is linkage mapping, QTL analysis, and genome-wide association mapping, that is GAVAs, employing different softwares like Carthagene, Fusion, and Tassel softwares. So over to you, Dr. Sujan, sir. I hope the participants will really get benefited from your talk and your experience.
Thank you, madam, for the detailed introduction about me. That's so nice of you. Before I start with the basic theoretical concepts and the practical aspects of that, I just want to start by showing the softwares that we need for today's session. First and the foremost, if you go to GitHub, under my name, there's a Quant Genomics Tirupati workshop. In that, I had all the PDFs or the presentations that I'll be talking today, and also the text files that would be, the input files that would be necessary for all the 3 parts of the 3 kinds of analysis. I had questions from few people about some of the issues with installations. So let me briefly go through each one of them. So the first one for linkage map, we'll be working with the software called as Carthagene. But before we install Carthagene, we have to make sure we have this active TCL, which is more like a Python or a Perl programmer. So we need to install this and install this. Just a heads up, this is how our thing looks like based on your architecture, whether you're using Linux or Windows. Here you have the Windows version. Go with the Microsoft Windows 1.3 beta, which was the latest version, was in 2013. This is the one. If it is non-free, it means it's for commercial companies, and since we're academic, it's free for us. So just go with this part. And in the presentation, I highlighted the same of the option which we will be using. If you click on that, it's a simple install. And remember, without active TCL install, this will not, even though it's installed, it doesn't work and gives us issues. So my first suggestion is first install active TCL and then install this. And for the second part, for the QTL mapping, we'll be using a software called as QGene. And for this, the prerequisite is a Java JDK. I think the present version is 1.7. We need to have that installed before we install this. So this is how the link I sent you looks like. So here you need to register, or you can directly hit on go to download page. And this is the one. So it's a simple jar file that's small and easy to, that needs no installation. In the third part, we'll be working with association mapping. Where we'll be using Tassel. And here for Tassel, again, the prerequisite is a Java version. Make sure that is installed. And for Tassel, this is the home page of the Tassel site. And here, based on the Windows or the operating system you're working with, Mac or Windows or Unix, you need to click on this Tassel file Windows 64-bit. That's the suggested one. And 64-bit are comparatively faster than the 32 bits. So I suggest downloading that. And some other softwares of interest would be TextPad, which is more like a notepad, but a few enhancements. Here is a link. And you need either a Microsoft Excel or WPS Office, which is a free version of Microsoft Office. So you can use that. And that's the same WPS Office I'm using for my PowerPoint presentations today. And as mentioned, all the slides that I will be talking today, all the raw data I will be using in this workshop will be available at my GitHub link. And if you have any installation issues, I think it's based on system Java versions and all those things. I suggest Googling a bit before you approach one of us. So with that, I'll just, Madam Amravati briefly mentioned about me. I'll talk a little bit about my work so that you'll get an idea that making me eligible for conducting this workshop. Today we have a lot of topics to cover. Might look a bit complex if you're using a linkage or QTL or association mapping the first time, but if you're already an user, you'll be able to relate with what I'm talking. So I suggest the presentation a couple more times in case you don't understand a few things. So today's the third day of the workshop. And as Madam mentioned, I'm a senior scientist working in computational biology at Hudson Alpha Institute. So far, in my last 17 years, I have been working with bioinformatics, biostatistics, and genomics, which are all interrelated. And based on the aspect we are looking at, we have genetic mapping, statistical genomics, quantitative evolutionary population. So I've worked with most of these things. This is my present Google Scholar page. I have 3,000 citations, H index of 26, and an I10 index of 40. And I only have 50 papers published, and still some are under review. Briefly, I have 17 years of research experience, 5 years of teaching experience, 15 research articles. I did supervise 15 students, more than 30 collaborations, and more than 10 workshops, and about 70 posters and presentations. I had a BSc in agriculture from Angro Bapatla, so I feel proud presenting at Angro Tirupati campus. And I had a PhD in genomics and bioinformatics at NDSU. So immediately after my undergrad, I went to NDSU to become a plant breeder. I was accepted in a corn breeding program, but due to some funding issues, I had to move to a different program and ended in genomics. So basically the place where I did my PhD, just to the environment a bit. It's in Fargo, North Dakota, border of Canada. Here the temperatures in winters are around -18 Fahrenheit, and we can feel cool days up to -40. The other 6 months is kind of summer, and it's the only season we have crops in. And in winter, we have about 50 inches of snow. And the coldest weather reported was in, if you take the top 5 places, Far West, one of them. And during my PhD, I worked on various aspects like linkage maps, in comparative genomics, association mapping, population genomics, demography, things like that. Just give me a second, my system is. And then in the same lab, I worked as a research scientist and an assistant professor, where I worked on beans and soybeans mostly. There I worked with the variant discovery, some introgression analysis, developing some of the methodologies, association mapping, and of course on the whole genome of common bean and population genomics, which was published in Nature Genetics, the first big journal. And then in 2015, I moved to Hudson Alpha Institute for Biotechnology as a senior scientist. It's a non-profit research organization. And we are proud to say we are the first team to offer genomic medicine, because right from the human genome sequence published in 2000, we had a lot of research done in humans for about 10-15 years, but it has to be used somehow, and we started the first clinic. So the idea is, whenever you have an undiagnosed disease, the parents and the particular kid comes to this clinic, we sequence their entire DNA and try to relate with known variants with known diseases or known issues. If not, we'll try to look for a few other factors. So the idea is, if the doctors give up their hands or if they're not able to diagnose what the issues are. So far, as far as I know, we have screened about 400 patients, out of which 2/3 were adults. And most of the symptoms we see are seizures, epilepsies, poor muscle tones, immune deficiencies, and others. The first question that comes is like, we always say our institute is a pioneer in genome research. The reason we say that is like, when the human genome was published in 2001 or 2001, Dr. Myers, who was then the head of Stanford Genome Center in California, contributed greater than 10% of the data, and a leading author on this paper. He's the president of our institute, or in other way, he's the one who started our institution. And he was supported by Jane Greenwood and Jeremy Schmertz, who assembled 3 human chromosomes right then, when there were no softwares, sequencing softwares, or no decent technologies softwares were available. And those 2 are my present bosses. These are the people I was talking about. On the left, Richard Myers is the director of our institute, and Jeremy and Jane are the faculty investigators. And basically, we have about 8 groups, and my group works with plants. All the other 7 groups works in humans. So basically, we work with comparative genomics, reference genome generation, population diversity in genomics, quantitative genomics, transcriptomics, and functional genomics. And so far, we have more than 150 plant sequences, which represent greater than 50% of the plant genome sequence. If you go to this is a website called as Phytosome, where you can have all the whole genome sequences available, and we did most of them, whether you need poplar, soybean, except rice and Arabidopsis. If you look at other crops, it's mostly rice. And as mentioned, we represent more than 50% of plant genomes, and we have many good publications from our lab. We have about 50 science and nature papers, and my work is mostly with variant discovery, working with population genomics, some epigenomics, time genomics, and functional genomics. And my areas which was poplar, pecans, or strawberry grass, sphagnum, beans, etc. And so far, I was able to publish a few papers in Nature Communications, Genome Biology, one in Nature Biotechnology as a lead author, and in Nature as a lead author. So these things I briefly talked about. So coming back, okay, I think this makes me eligible to talk about what I'm talking today. So before I go to the linkage map development or using Carthagene, I just want to talk about a few concepts of linkage. So basically, our genetics starts with Mendel's laws. You know, we have three laws. One is the law of dominance. So basically, when you have two homozygous individuals in a crosses paired with contrasting traits, the F1 hybrids are dominant, and the traits that don't appear are called as recessive. And in the second one, we have law of segregation, where we have two copies of the genes, where each one of them enters the sperm cell, and after fertilization, you get heterozygotes, and further fertilization, you get gametes occurring in ratios, which we call it as the dihybrid ratios. And then you have the third law of independent assortment. Sorry if I'm being too fast. I have a few things to cover today. And since this is all basics, I'm not going in detail of this. And the third one is the law of independent assortment. Basically, it says when we have two or more different genes for a pair of traits or characters, they assort independently into gametes and such that they form into nice ratios. So, for example, you know the yellow, green and wrinkled piece and yellow and green piece. If you look at the dominant and the recessive traits, we have a dihybrid ratio of 9:3:3:1 based on this law of independent assortment. But there are situations when this law of independent assortment doesn't work. So the main two reasons are either by chance, meaning the meiosis or whatever the natural conditions did not happen properly. And the second one is linkage. Linkage is where when you have genes or alleles on the same chromosome and are very close together, then there is no independent assortment of the alleles. One thing to remember is this law of independent assortment was framed before we know what meiosis is. So what exactly happens during meiosis is haploid gametes are formed from diploid cells, and then the independent assortment of genetic loci happens. So when you take two genes or two loci in the same chromosomes, we have crossover or recombinations, which lead to various combinations of alleles. So linkage is basically when two genes or two loci are linked, and independently they segregate together in a population. And once linkage is based on the frequency of crossing over. If you look on the right, this is what the crossing over is. So it's basically reciprocal exchange of chromosomal segments. And then our main topic for the day is linkage map. So based on this linkage or the recombination events or based on this crossover, we do develop a linkage map. Basically, this is a map based on recombination frequencies between genetic markers during a crossover of chromosomes. Looks complex, but let's make it simple. Basically, whenever you have a population, say we have an population or a real population or a backcross population, suppose we have a sample size of 100. It's not a single crossover that led to this 100 cells. There are multiple crossovers, multiple recombination events in all these 100 samples. So trying to understand where they are, where the recombination events are, or how many samples did this recombination occur is what we do linkage map. So on this linkage map, we have a unit called as centimorgan distance, which is also equivalent to recombination frequency or the crossover rate. So it's basically, it says how many crossovers or how much recombination occurred for that particular cross. So if the frequency of recombination is high, it means the two genes are apart. Let's suppose we have a centimorgan distance of 1 centimorgan, then we mean to say we have one recombinant. when we say 50 centimorgans, we have 50 recombinants, meaning if they are far enough, there is a higher chance of higher recombinations. And definition between two genes equals one half the average number of crossovers. But let's not worry about the definitions for a bit, because as I mentioned, we'll be concentrating more on the practical aspects. So the first question that comes to our mind is, why do we need linkage maps? They started linkage maps from early 1980s, but even today in the next generation sequencing, do we really need linkage? The answer is yes. The main things that we use linkage maps today are for genome assembly. Even though we have whole genome assemblies released, without a linkage map, we cannot assemble them. I'll talk in a minute. And then we have map-based cloning, and then we need for studying genomic structure, evolution between different organisms, which is also called as synteny. And then for QTL analysis. QTL analysis is nothing but identifying genes or regions on the chromosome that are associated with the trait, so that we can identify those genes, do some functional studies, or identify those markers and used for marker-assisted selection. Let's talk briefly about each one of these. When you talk about genome assembly, so basically we have a fast or raw DNA reads. Previously it was 150 base pairs to some 200 base pairs max, but with advanced PacBio and BioNano machines, we have up to a few kilobase pairs, 5 kbs, 10 kbs, and things like that. So you have those reads, and then you develop them into contigs. Contigs are nothing but these reads. If they have overlaps in the ends, they overlap and merge into a single thing, which is called as a contig. And these contigs, based on certain other criteria, which we call it as gap sequencing and many other things, we develop them into larger scaffolds. So to put it in precisely, our reads are a few kilobase pairs, let's suppose 5 kilobase. Contigs are 5 kilobase pairs, still a few megabase pairs or hundreds of kilobase pairs. And you have scaffolds where you have multiple contigs assembled together, but you don't have the sequence. You fill them within and get scaffolds which are a few megabase pairs, up to a few megabase pairs. And then using certain other things, we can do gap filling, and then we develop into long chromosomes. So the question comes is, how do I organize these contigs into scaffolds and scaffolds into chromosomes? For that, a linkage map is necessary. Let's suppose I have two contigs, and I have marker 1 on chromosome 1 and marker 2 on chromosome 2. So I can say contig 2 comes after contig 1. So without a linkage map, we cannot determine the order of the scaffolds or the contigs, or we cannot build into chromosomes. The second part I mentioned, second advantage is map-based cloning. So basically, you start with some QTL analysis or intracrest analysis. This is a classic example in identification of a disease gene in wheat. So on chromosome 5, they identified a small region using initial SSR markers, and then they took other markers from the same region and tried to pinpoint it, and went down multiple steps, and they identified it to be a NBS LRR domain containing this gene right here. This is the gene that is responsible for trait mediation. So this is years of work, if I remember correctly, from Justin Faris. It was about 15 to 20 years, because it was a combination of multiple crosses and multiple marker systems. And I mentioned about marker-assisted selection. So basically, in a regular pedigree method, you have two parents crossed. You have your F1, and your F1, you grow it in the field. You do phenotypic screening, select a few, grow them again, cross them again. You continue till you get some F7 or F8 generation, and then you go with preliminary trials and advanced tail trials. Whereas when you have marker-assisted selection, the advantage is here, instead of phenotypic screening, will be a marker-assisted selection. Only if we identify the marker that is responsible for that trait is identified. And compared to on the left, here you see only few plants are grown in the field. So with this, you save a lot of money and time phenotypic again. So, and that occurs during all the stages. One advantage is a less amount of time and labor necessarily to phenotype. because you need multiple replicates, multiple locations, and many other things for release of a variety. And the other thing is sometimes you can even uh, remove one generation out, or remove in the sense like you can avoid growing them in the field. For example, let's suppose I have an F5 population. If I grow in the field, take all the phenotypes, say for example, plant height, it takes me about 5 to 6 months at least uh, for an annual crop to show up and give me the phenotype I'm looking for. But for the same 5, 6 months, I could use a marker system and use marker assisted selection and direct within a week or less than a week based on the technology we use. Within less than a week, I can select the same 100 plants out of 1,000 plants. So that saves generations and also the time and labor requirements. And the other thing I mentioned was the genome evolution. So here we have some chicken, zebrafish, and blue tint. So if you look at these three genomes, the same markers in all the three systems, the thing to notice is there is an inversion here, and there is also an inversion here. So inversion or a translocation here, and here we have an inversion. So these are the kind of things we see when we have molecular maps or linkage maps. And one other advantage which we will be talking about is the QTL analysis. So basically we have some genotypic information, some genetic map, and some phenotypic information. We try to identify the region in the genome which is responsible for that in a minute. So basically for genetic linkage mapping, the main steps are we need to develop a mapping population, which can take years, and we need genotyping or a markers for this population. Could it be SSRs, RFLPs, RAPIDs, KMRs, SNPs, could be any system. And then once we get both of those things, we will be constructing a linkage map. The first software that was developed for linkage map development was MapMaker. And now I use Carthagene, but if you want to do advanced and more things, the QTL cartographer is a very good thing, and RQTL is one other good thing that does really decent work on linkage maps. But I prefer Carthagene for training purposes to get the concepts easily because there is no commands. In RQTL, you need to do some commands, and here it's just click of button. So first time users, it will be really easy and it's really well established software. So when you talk about mapping populations, so basically you take parents which are contrasting for trait of interest, meaning if say, for example, I want to understand the genes responsible for plant height. So I need to take a short plant, tall plant, cross it, and develop a segregating population. So you have F2s, double haploids, back process, recombinant bread lines. These are the common for linkage mapping. F2, you're talking about two generations from parents, which is reasonable. When you're talking about recombinant inbred lines, it's 7 to 8 generations. So you're talking about 8 to 10 years. And then you have other methods. So the first question, recently you might be seeing papers using F1 population for linkage map development. Yes, it is possible if it's a highly heterozygous species. And that one we only know when we have the whole genome sequence available, or at least some sequencing information available for that. And also recently we have been looking at magic populations other than the crosses I mentioned above, to capture more recombination events. The problem with this is, for example, if you take common bean that I work with mostly, in real lines, after 8 generations of recombinations or crossovers, hardly any population, you get hardly 1,000 recombination events, not more than that. But if you take magic populations, here you have eight ways and 4 ways. So basically you have A by B, C by D, E by F, G by H. So crossing them, this an 8-way cross basically. So in the final thing, if you look at the 8-way, we expect a higher number of recombination events. As an example, in bean I mentioned, which is say 1,000 crossovers or 1,000 recombinants overall. And here we see about some at least 5 to 10 times more than that. Of course, this is based on the parental differences and other factors. And one other thing is, so far map making was restricted to diploids, and in polyploids we had some issues. But polyploids are quite important because most of the crops we see today, like canola, grapes, which were sweet tomato, strawberry, potato, alfalfa, whatnot, most of these are polyploids. You had two kinds of polyploids: auto polyploids, which are within a species, they are homozygous at every locus, and you have yellow polyploids, which are raised due to hybridization of two distinct species, example wheat, for example. These polyploids are in general have hybrid vigor and better adaptation, and they have gene redundancy, meaning a gene is present multiple times, giving it a buffer to do better. So when you're considering tetraploids, they can segregate in F1 progeny. So here are the different parental genotypes that are possible. If you look here closely, in diploids, we were mostly talking about homozygous parent 1 and homozygous parent 2. But here in polyploids, in F1 progeny, if you want to map it in F1 progeny, we look for even heterozygous parents. So you have different combinations where you end up having disomics and tetrasomics. It's in allotetraploids, it's disomic, whereas it's tetrasomic in autotetraploids. And the second system the requirement is using markers. So with a dominant marker, you have two states, presence or absence, for example, whereas with codominant markers, you have a big, small, or heterozygous condition. This is okay. This is, for example, parent one small band, parent two large band, whereas your F1 is, you have both the bands. And the marker systems could be SSR, SNPs, GBS, NGS, multiple Lillian chips, or any other things, and even KMers. KMers. Recently, you might be, I think in the last 1-2 years, there are a couple of papers out using KMers for linkage maps. Basically, KMers are nothing but a short fragment of sequencing. For example, if you have a sequence of A, B, C, D, F, G, H, so if you take KMer as an example, and you're taking a three-mer or three base pairs, but in general, we take a 24-mer or a 72-mer based on the species, heterozygous, polyploidy conditions, and everything. So we search for these things. For example, in this, we have ABC, BCD, CDE, so on, a continuous walking strings. So these are our KMers. ABC is one KMer, BCD is one KMer. So within each species, within each sample, we look for these KMers, how many are present, present or absent, and we make a matrix, and we use this matrix to make a linkage map. And in case of polyploids, so based on the parental genotypes or the parental alleles, we had different types of markers called as null markers, simplex, triplex, duplex. So you might be hearing these words a bit if you read polyploid genetic mapping paper saying that we use triplex markers or we use duplex markers, something like that. When you want a linkage map in polyploids, I suggest not to use Carthagene. You can use Polymap or Tetraploid SMP map or NetGivas or some of them. And one other common terminology we come across is map saturation. See, when we develop a genetic linkage map, you might end up having the distance between markers to be, say, 5 centimorgans, 3 centimorgans, so on. So when you talk about 5 centimorgans, for example, there might be 200 genes. So if your QTL region is within a 500 centimorgan region, you're looking at 200 genes. So it's so hard to identify the exact gene that we could clone, or that we could do CRISPR, or that we can do any functional studies. And in general, we use near inbred lines to identify, to narrow down these intervals. So basically, it's, I mean, I don't want to talk more about details. For example, in soybean, we have Williams 79 and 82, where they differ only in one regions, where the phytoproteins are introgressed. So from 5 centimorgans, we use more markers, and we try to narrow it down to say 0.5 centimorgans, which represents say only 20 genes or something. And as you are aware, these genes are not equally distributed on the genome. So there's something to keep in mind. And one other concept that you would be hearing is the map merging. These are the 3 different softwares that do map merging. So basically, say for example, I have a cross A by B real population, and they have a molecular map or a linkage map. And someone else within the same species, they have a cross C by D, an F2 population, and they have a molecular. But let's assume both of us were using the same marker system. In that case, we could merge both of them and make a decent. The advantage with this is, I mean, this is not highly accurate, but this is reasonable. The advantage with this is you will be able to accommodate more and more markers. For example, if I'm looking for some price variation in say common bill. If I only consider say SSRs, maybe I have some 2000 markers. If I add more marker system, if I consider more populations, I can have more markers, say 10,000 markers, and at the same time, I can narrow down my QTL region from say 5 centimorgans to half a centimorgan or even less. So this is one. For this, you can download all linkage maps available for that particular species. Feed into merge map. It's the one I used frequently. It was a really good job of calculating, doing all the statistics behind to get our final thing. For example, in the map A, we have only 3 markers. In map B, we have only 4. But when you come and see, you have only 6. When you combine, you're able to get 8 markers, which are comparatively closer to each other compared to the individual maps. So some of the practical aspects or some of the steps we need to do for linkage map is first step is data cleaning. For that, we need to have, I'm talking about diploids only and the simplest methods. So parents should be polymorphic homozygous, meaning if parent A has say capital T, parent B should be small t. But in our case, we'll be using A and B. Parent A should be A, parent B should be B, or the other way around. And they should be morphed such a way that parent 1 is always an A allele and parent 2 is always a B allele, such that it calculates the recombinations more accurately. And we need to duplicate marker names, we need to samples based on missing data, and we need to check if they meet the criteria. For example, if I'm using a real population, are they close to 1:1 ratio? If I'm using an F2 population, are they close to 3:1 or 1:2:1 based on the marker systems we use? So is it close enough? We need to look for, and we need to filter out the unnecessary markers, because you might end up having a marker which is all A's, which is all parent 1. So there is no use, there is no recombination in that marker. So those are the things we'll be removing when we check for ratios. And whenever you're feeding your data into the software, we need to make sure there is no parents data. This is an outline for Carthagene. I'll talk about in a minute. We'll make what it is. So basically, there are 3 lines on the top. The first line should have data type, some information. That X is nothing but whether you're using RILs or interprocess or back process or whatever. And the second line, we have 3 numbers. The first value is the number of samples. The second value is the number of markers. And the third one, if you have any trait values, which we don't in general. And this is an example. Sometimes you can have optional symbols as 1, 2. If you have, instead of A and B, if you have 1, 2, so write this way. And that from then you have the marker information. In this particular example, there should be a star in front of your marker name indicating a name, a space or a tab, and then you have AB dash means missing. This is the genotypic data of all the individuals. And these are some of the useful commands. I included them in the slides. I want you to have a look if you're working with them. Let's not worry about all this because these are typing commands. Today we'll not be dealing with the command version of it. Okay. The main steps would be, I will talk in a minute, we'll be using a load and select the file, we'll look for the information, then we'll be merging all the markers if they are the same, meaning you might have 2 markers, but the data is exactly the same, meaning there is no recombination between them. If we narrow down the number of markers to be used for analysis, that makes our analysis faster. So sometimes we merge the markers. And we can config for the different algorithms. Then we identify groups, and for each group, we repeat 3 steps where we identify the group, We use some algorithm, we flip and polish. About this, I'll show you in a minute. And then copy the output into a file. And I have included all the steps for data cleaning that I use. Let's do one by one at all of these. I'm just looking at the messages in case if I need to enter GitHub link. Okay, sure. Janki Ramaya, I'll try to talk close to the computer to avoid my voice breaking. That's the link where we could get all the files. So the files I'll be talking about right now is the linkage mapping raw data.xlsx. So let me open it. I'm just making a copy of the original file. I'm trying to paste side by side so that we could simultaneously look what I wrote and what is being done. So this is a real population. I have on my right is an Excel file with all the real population. So there I have a marker name, parent 1, parent 2, and if you set the real population. So there are about 91 samples. This is some rename data, original data of error boxes where we changed the sample names, the marker names, and a few other things. So the first part is, I said, just highlight the parents. In my case, I had P1, P2, but you might have some names, some Williams by some something, whatever the names are. And in the next step, I said, look if the parents are polymorphic. For that, I'm just inserting a column on my left. Here I'll be talking about a few Excel functions. I don't know how much of you are aware of them. So the command for that is exact. This one, comma, this one. So it's saying, it's comparing those two. They're not the same. So it gives a false. So all are false. So no issues. So parents are polymorphism for all the markers. In case if you have say both A and A, it gives you true. Meaning in that case, you have to remove that first marker out. We're done with first step. And the second step we want to make sure is parent one is all parent one is an A and parent two is an always B. So for that, what I would do is select all data, sort, custom sort, and here let me sort by parent one and then by parent. So AB, it's all doing good. But if you see here, there are a few markers where parent A is B and parent 2 is A. So this is something that has to be taken care of. Just highlight it with color. And then there you have SNPs. These were randomly created. So if you look at SNP, in case of SNP, you have it as ATGs, it's not A's and B's. So you have C and A, you have G and A, you have T and A. So they have to be replaced as well. I highlight to make sure that is I'm doing the right cells. So this is the first set of cells which we'll be doing. So here the idea is these should be swapped. So for that I use a method using a third variable, shift to right arrow. Menu, edit, or replace. I'm converting all my A's to a third alphabet, say Z. Replace all. Then I'm converting my B's into A's and Z's to B's. If you look here, see the highlight ones, even here we got as A and B. And now let's replace this. A becomes Z, C becomes A, here Z becomes, I just want to do the other version, so B. A becomes a Z. G becomes an A. Here a T becomes A becomes a Z. T becomes an A. Z becomes a B. So with this, we got everything into the same format that we're looking for. So that completes part one. Then we need to look if we have any duplicate markers. So for that, I don't know if the same exact function. So I'm just sorting it by data sort marker. I'm just comparing this cell, which is B2, with above cell B1. So if they're the same, it should give me a true value. So it's almost false. We don't have any duplicates. I'm showing the steps so that, if you in real time, if you do it for your data, it will be something useful. And the next step is we need to count for missing data, count alleles, estimate the chi-square. So let us do all of this. The first thing is missing. Let's count B, A, B, expected A, S. So the functions are is equal to count if the range excluding the parents. It's a dash within those cells, just count it.
Excuse me, sir.
Yeah.
Sir, then what score we will give for heterozygote, sir?
Good. This is a real population. So I had only ABs, but if it's a heterozygote, we give it as a H.
Sir, I have another doubt that is it compulsory that RILs will not have heterozygotes at all, sir?
This is the eighth generation RIL. So
Yes,
I
sir.
mean, after 8 generations, you're talking about 99% of homozygous condition, right? I mean,
Okay.
like 50, 75, 82.5, so on, right? So
Yes.
it goes to 99%. And so we assume those very low number sequencing errors.
Okay. Okay. Thank you, sir.
So there are 7 missing in this. Again, so total, I said like we had 101, so that matches it. Okay. So this is just for this marker. I said expected A, expected B because this is a real population and my ratio expected range is 1:1. So just for the data that is present, excluding missing, just an average name and just a simple number. So that would be, I would say, floor because we don't want the decimals. This one plus this one by 2, 1. So we have 94 samples that have data. So our expected ratio is 1:1, right? So 47:47. So to get this, I used a formula called as flow. First, let's do an average. 54 plus 40 is 94, divided by 2 is 47. This is a good number, but in case if you have decimals, you get 0.5 or something, right? So to avoid that, I used another function called as flow. meaning it rounds to the lowest number with just one whole number without any decimals. So we had this expected ratio. Then let's take chi-square test, chi-test. It's saying actual range is this one, and our expected range is C2. This is the p-value. So let's extend all this formula to everything. Done. The next thing is I want to sort it, but these are all formulas, right? So to take the formulas out, what I do is copy, copy, and then go to paste the same values. That way you lose all the formulas. You lose all the formulas. Now let's do some sorting, removing unnecessary things. We did the chi-square test. So here in the slides, if you look at, I included all the formulas, all the cells, everything. Data, sort, custom sort. First, let's look by missing data. Largest to smallest. See, out of 101 individuals, we have 89 missing. So we could remove up to, say, let's say 50, which is 50% roughly. 50% of the samples, if they have missing data, we're removing that data. And then let's sort by chi-square value. So let's, if you take chi-square is a point where your expected ratio and the observed ratio exactly match 40:40 or so on. So let's have a cutoff of something, say 0.01 maybe. Yeah. So I will remove all these that don't match our criteria. So finally, we are left with 91 markers. And we don't need any of this information. We don't even need parents. So I'll just copy, Ctrl+C, the new sheet, Ctrl+V, remove the parents. Now the thing is the data, we have the data that is necessary for input into a map making software, but this is not exactly the format. So we don't need, without the marker name, let me select all the data using Ctrl+Shift buttons, Ctrl+Shift right arrow, Ctrl+Shift down arrow, Ctrl+C. And as I said, like there is a software called text patch. It's more like a notepad. The beauty of this is it has large more features than the regular one. Here, for concatenation, I need to remove the tabs or the spaces in between. So for that, I'll say search, replace, /T, meaning it's a tab. In programming language, we have /T, /P/ and so on. T is a tab, / and is a new line. So in our case, we have a tab between. I'm saying remove them and do nothing. And make sure your regular expression is on. So I have highlighted all those here. Replace all. So it removes the tabs and gives you as a single character, single string. Now copy back, Ctrl+A, Ctrl+C, back to our data, and copying here. Ctrl+V. And here, from here, I'll just copy the marker names. Ctrl+C. See, you have these issues because they start with a dash. So we need to clean up those issues. Select column B, format cells, change it into text. Okay. And if you see here, just remove this equal symbol. See, since it considers initial values as numbers, it takes that as a minus. So it includes an equal to symbol in front. Edit, replace, equal to with nothing. Did not work out. So we have all that issue solved too. So this is our marker data. Control C. So you remember we had 2 lines on the top. Okay. This is the same input file for even a map maker. Data type, array self. Instead of array self, you can use RAC and many other crosses based on the cross you use. And then we have the number of samples, which is 101. And we have the markers 91. And a 0, no quantitative traits. Remove the extra lines. Save. Plugin input.txt. Save. Okay. So this completes our input file making. So if you have everything installed properly, just looking for that app should do it. So this is the Kartaji. On the top, we have some buttons that we use, and on the left, we have some buttons. The first command, if you look here, so basically, if you look at, there is a help. You have a help button here where you could find all the commands that we use. You could search for them. And these are the buttons, but at the same time, you could type in your comments. For example, I can type in which is above here, which is right here, but I can type in here and change the options. So if you click on the button, it works with the defaults. So the first one is you have to load or this or it is saying syntax server marker 0818. A second, let me. I don't see that marker name at all. They had a different before the workshop. I was able to load this and run this. Your second marker is showing the issue. Just a minute. find the problem. Sorry about that. Sincerely apologize for this. I checked it in the morning and everything looked normal to me. Was able to even run a couple of linkage groups. So I have another file that I will upload. So basically, it should serve our purpose. So this is from I instead of the data file I provided, I'm able to get the other data. So, but the procedure remains the same. I'll try to see what the problem is with our data file, and by the end of the day, I should be able to do that. Coming back to our steps. So the first one is load, where we can load a data set. And the second one is once we load a biological data set. So if it shows how many are loaded, it's saying this is an RSF population where we had 122 samples of 101 markers. It meaning it's meaning is it's loaded successfully. And then if you go with data info, it will give the details. You go with data info, it's saying the what's the data type is, how many markers, individuals, and what was the file name, just to get an understanding. I mean, you can click this button anytime in the middle of your work so that you'll be able to see that. So here, these are all the markers, and this is from Arabidopsis. As you know, Arabidopsis has 5 chromosomes. So basically, you have a few markers from each chromosome. So you would expect 5 chromosomes if they're decently distinct enough. But if they're really far enough, you'll have more than 5 linkage groups. So a linkage group here is nothing but a set of markers that have recombination frequencies less than 50 between them. So to do identify that, we'll go with loci, identify groups, and there are 7 groups basically. You should give this. I think there's some problem with my TCL or my Windows version. Need to recheck that. So basically, there are 7 groups. So 1, 2, 3, 4, 5, 7. And if you look 1 and 2, they are just a single marker. They don't fit anywhere. So you have 5 remaining groups, each one probably corresponding to a single chromosome of Arabidopsis. So once you identify these groups, we need to select one group. So for that, loci, select a group. Let me select the last one, for example. Okay. So it's saying marker selected. These are all the marker numbers in that. So it selected all those groups. Then if you look at loci, distance to point, if it gives a big matrix. Basically, the idea is if the distance between 2 markers is the same or it is 0, then meaning it has no recombinance. So here, say for example, this 0 is the one I was talking about, meaning this marker G1N838 and G388 co-segregate. So let's not worry about that part for now. Let the software figure it out. So this is the basic. This is the base nice map. D nice map D is the basic algorithm to identify the order of the markers on the chromosome. So it's saying this is the likelihood, and if you go with the details, this is the genetic map. But that doesn't end our thing. We need to do a better algorithm where we are confident about the final lot score. As you are aware, based on our requirements, lot 2 or lot 3 is the minimum lot score for a genetic map. So let me hit on build FW. So it does some combinations, some steps. Let's not worry about that in detail. So this is the final map. So if you look at the distance between T3843 and G448 is 5.5 centimorgans, meaning there are 5.5 recombinants between these two. So on, it gives the total map is 127. You can either do a Haldane function or a Kosambi based on your requirement, but Haldane is the both are used on and off, so it doesn't matter what you really use, but just stick to one. So this is able to place 14 markers with a total distance of 1 or 2 centimorgans. So I can take this, Control C, Control V. So now let's select another group. Loci, select a group. We selected the last one before, right? Let's select group 6. Okay. The first one I would do is directly you can hit the build FW. Hit it. Only 3 markers are included. All others have been excluded because this is build FW is highly strict in the lot terms. To set those things, go with the loci config. For example, so go with such config, and this is the algorithm I have been using. It's saying keep threshold should be 3 and the add threshold should be 3, meaning once a marker is added, it should have a log 3 itself, and the overall should also have a minimum log of 3. So I can reduce it to say 2. Okay. So if I go again loci, select a group, the last but one, sixth one, build FW, detail. So here we had 7 markers now, but this is only at a log 2. So based on your requirement, you can do. So the same way you will be doing for all the 5 linkage groups. Let me redo it again. So you'll get an idea. Save the configuration file now. So what we did was, it's so simple, Karthajin, if you know the steps, it's just a click of 5 to 10 clicks, you can get linkage group in 5-10 minutes. But I'm resetting the session to remove, clean up any issues we have. So load. Right now I did. Okay, I loaded it. It loaded successfully, no issues. If I want information, I click with the data info. It gives me the information on the file type, number of markers and individuals. First thing I do is a loci, identify groups. It gives me the groups, and I see there are 5 reasonable groups. Now loci, select a group. I'm selecting the 7th one. Okay, hit build FW, detail, copy paste. If you want a graphical plot, you get something like this. If you want a Pareto, okay, it's not sure. So we're done with 1. Now I already copied this. Loci, select a group, 6. Okay. Build FW, detail. Copy. So on and so forth. Next, loci, select the group, group 5. Okay. Build FW, detail. Only 3 markers here. But if there are other algorithms that are better than these 2. So for example, let me loci, select a group, 7th one. Okay. Build FW. As you see, we had 14 markers, right? So once you get this, the next step to confirm this is called as a flips, polish, detail. It's the same 14 markers. Sometimes what happens is it flips a few markers that and this and does some permutations and combinations. So basically the main steps we have is loci, select a group, loci, let's do the third one. Loci, select a group, build FW, flips, polish, detail. 4 buttons, and this is your final map for that linkage group. There are another things like annealing, taboo, genetic, which takes a bit of time, and it uses a different approach of likelihood and everything. Over time, we found this build framework map to be reasonable for getting linkage maps. So once we have this, for example, this linkage group, we're expecting 5 linkage groups since this is Arabidopsis. So you have the distances between all these things. That completes our linkage map thing. And now it due to this data, but I hope you got what we are talking about, the linkage map. So I wrote build FW. You can change the configurations. The best map is the last in heap. So you can just hit the detail or the best print if you want commands. So I included each and every step possible here. And the second aspect for today's class is QTL analysis. I briefly want to talk a couple of minutes about this. So basically, you know, quantitative traits and qualitative traits. Qualitative traits are discrete phenotypic scale, like for example, seed shape, which is round or wrinkled, seed color, gray or white, so on. Whereas quantitative, they're polygenic or to multiple genes at the time, and you see continuous variation. And QTL analysis refers to quantitative trait analysis. The one on the right is the one we're targeting that. Mapping is continuous variation, and they do not fall into discrete classes. It's basically controlled by multiple genes, hence the polygenic or multifactorial. Our idea is to identify those multiple regions that are responsible for trait variation. So for example, this is a kind of a normal distribution that we kind of look when if it is a quantitative trait, and most of the traits in nature are quantitative. So these quantitative trait allows refers to regions of the genome that are associated with a quantitative trait. It could be a gene or a cluster of linked genes, but in general, these are controlled by multiple genes, each segregating according to Mendel's laws, and these are affected by environment. Meaning, it's the same set of plants. If you grow in a single plant grown in different environments can perform differently due to this environmental conditions. And since it's controlled by many genes, the individual effect of each of these genes could be small. And these are subject to epistasis or a combination effect. For QTL mapping is identifying the regions in the genome that are responsible for this trait variation. So for the, again, this is only taken in case of mapping populations. Just like a biparental left to population, RILs or whatnot. So the main objectives are identifying the regions, what is the effect of the QTL, is it an additive effect or what the effect of that is, or a dominant effect, and how much variation or how much change in variation, trait variation is causing, and which allele is the favorable effect is the target. So for this, the prerequisites are we need a good cage map, which kind of we develop, a segregating population that differ in traits of interest and phenotypic values. There are different methods: single marker mapping, simple interval mapping, composite interval has different statistics, and based on our requirements, we do it. Single marker analysis is where we don't have a linkage map, but we just want to know the markers for marker assisted selection. And simple interval mapping is it takes intervals between those markers. I'll show you a bit. Electron analysis is it does not require a linkage map. So basically, it does an ANOVA or a linear regression to identify if the marker class is causing a difference in phenotype or not. So for example, here, if you have a non-significant test from ANOVA, that the marker is not significant, or the QTL near that marker is not effective. Whereas in the case of the right, the means are different, hence there's a marker at or near the QTL. So the limitations are, we don't know the positions. So we go with simple interval mapping. Basically, it takes advantage of cage map. So if you have, say, marker 1 and marker 2 at certain positions, for example, this is at, say, 5 centimorgans, this is at 10 centimorgans, it assumes QTL every 0.2 centimorgans, calculates some likelihood, and gives the, instead of just saying the QTL is between 5 and 10, it says the QTL is somewhere between some 6 and 7 or 6 to 6.5. So it narrowed down the position. This is simple interval mapping. And here is an example. So we have M2 and M3 at say 5 and 15 centimorgans. So let's assume you have QTL at 10 centimorgans. That was not sequenced. But here, every 2 centimorgans, it does a walking distance and tries to identify the QTL at 10 centimorgans. So if the statistical method used for this is a maximum likelihood effect, let's not go into the details of it. We need a log of minimum, which means that it is 100 times more likely that a QTL exists between this interval. Log 3 is better, so which is 1,000 times more likely. So the log score is generally a measure of strength of evidence of presence of a QTL. This is an example of a simple interval mapping QTL. But here the problem is the curve could be spread over multiple centimorgan differences and could lead to false positives. So there is another method that we'll be using today is the composite interval mapping. Basically, it's same as simple interval mapping, but takes cofactors. Some of the markers as cofactors. Cofactors control for confounding effect. Meaning, for example, let's take one marker on chromosome 1. You have they are related to 5 other markers and 5 chromosomes. The problem is when you do simple interval mapping, you see 5 peaks, but in reality, it's a single peak, but one is controlling the other one based on certain LD principles or whatever. So in that case, when we use marker 1 from chromosome 1 as a cofactor, in that case, all the other 4 ones show to be non-significant. So that reduces some false positives. So let's not go into the details. So this is simple example of a single marker analysis, simple interval mapping, and composite interval mapping. If you look between simple and composite, see in composite, it's a reduced distance or a reduced peak. Whereas here, if you take this as the cutoff, it extends this much roughly. So in case of polyploids, it's the same as diploids, but whereas it needs a different software. Tetraploid map is one if you're using a tetraploids. So with the QTL mapping, we identify nodes that are responsible for trait variation. These are from biparental populations. These are advantages over the mutant populations. Mainly, it can only identify loci that have larger effects, say 10%, 25%, or so on. But the main other limit is specific to that population. So if you have a cross of A by B, the QTL identified is specific to that population, not necessarily significant in the C by D population. So you have a genetic map and a physical map. So here you have, so your genetic map might be close over, but your physical map is, sorry, this is a genetic map, centimorgans, but on physical map, there could be close because you have gene-rich regions. So your QTLs are mostly from those gene-rich regions. Coming to our software today, QGene input has 3 sections. The one is the header, which contains study name, for example, plant height, a mating string type, genotype symbol, some information, parent 1 and parent 2. And these are the various strings that could be used for backcrossing, double haploid, selfing, so on. And we could use a combination based if it's a complex cross. And for the parent 1, homozygous caplase, parent 2, homozygous small a, heterozygous small a, so, or the dominant marker types could be written this way. So the genotype symbols in our case, for example, we have only A and B because it's a real population, but F2 population we have A, B, H, small a, and we can a small b if we want to have. I mean, based on the population type which we have. And the second section is the where we have chromosome, marker, position, and the alleles. So the centimorgan position ordered. So this is the one we'll be having. And the third one is the trait data. That type, the second column is the trait, this one right here, M. So basically there are 3 types that are accepted in QGene. One is nominal, ordinal, and metric. So nominal is discrete classes like color of flotilla, orange, red. So you just give 1, 2, 3, 4, so on. Ordinal is based on order, low, medium, tall, for example. Or disease, highly susceptible, not susceptible, resistant, highly resistant. So these are based on ordinal. In that case, we give ordinal. And the third one is a metric, where you have a numeric data. So for this, coming to the questions before I go to QGene. Well, yes, MapMaker and Carthagene need a star for sure, Amit. Bhavya, missing value depends upon the population size. We need a population size of minimum 100. I would say if the missing limit is only 10, that would be great. But since our we can go until 25, 30 based on the number of markers. Say, for example, if you have a next generation sequence, 10 million markers on your population or a million markers, it doesn't matter if you lose 90% of them, right? So it's based on that. And Gulab, sure, I will send by the end of the workshop. For Q gene, it's a double click, it's a jar file. In case if we have issues. Okay. In case if we have issues, we have to go to command prompt as mentioned in my slides here. I mean, it depends upon the jar version, your computer operating system, the configurations, all those things. So you have to open command prompt, you have to type in java.jar program name. So again, here the first step is file, load data that is provided in GitHub link download page. All files, so here I have two traits. These are the chromosomes, about 10 of them. And this is my marker data for the homozygotes and missing. You have two traits for the chromosomes, marker data. So first let's look for analysis, trait analysis. So we have Xantho 47, which is a bacterial infectation. So this is the curve. Well, as long as it's not discrete or multiple, close to normal or uniform or reasonable. So this is a reasonable thing to look at. And if you want any transformations, you can do those transformations right away. Histogram normality test. The Windows closed. Okay, here, for example, this one is showing all the statistics, all the normality tests, case test, Shaprobel and whatnot. So you can check whether it's not normal or not. But seeing the histogram, we know it's not normal. So in case you want to save this data, you can directly go with file, export text or export image, import or file and save this particular histogram to be used in your thesis or whatnot. And our main step today is QTL mapping. So let's go with QT analysis, QTL mapping. So the advantage of QGene is you have all the options and you write all the different types of algorithms or the analysis types. And here on the left, you have all the chromosomes, 11 chromosomes and 2 traits. First look like Xantho 47 is the trait. And let me select all the chromosomes. Let us do a simple interval mapping. And for that, I will take a lot. And we set minimum lot 3. It's lot 12. So meaning there is some peak somewhere. Here it is. Chromosome 7 starting from here to, so let me select on the 7, for example. So if I take 3 starting from here to here, this is our QTL region based on single marker analysis. And we can have what the additive effects are, what is the R square for each of the markers. So, and it seems this particular markers has 53% variation explained with this. But this is not the one we are interested. Sorry, I clicked on simple marker, it should be single interval marker. So this is the lot, this is the R square, 553%. But let's go to the final important one that we are looking at directly. Composite interval mapping using like. So for that, select chromosomes, lot, the same like before. But if you look at the peak here, is this. So let's go to options, select cofactors, stepwise cofactor. Okay. See the QTL peak, it reduced from say 5 to 10 centimorgans to all the way to 1 to 2 centimorgans. That's the beauty of using cofactors. The first thing that comes into mind is, I mean, in our case, we have more like a single pillar, but what should be the cutoff? So in that case, we do resampling permutations. It's 1000 default. Let me do 50 so it's faster. Start. I suggest doing a minimum of 200. So it says I should use a minimum of 5.37 as the cutoff. So coming back to our data, so whatever is greater than 5.37 is my QTL peak. So how do I remember this? For that, we have options called as, let me reverse the video, looks better. Go with file, export text to clipboard, or you can directly save to file. But in my case, I do this. And this is the Lord's course, if you remember correctly. So it's in chromosome, the locus name, position, composite interval mapping. If you remember correctly, on chromosome 7, we had our QTLC of 0.2, 0.1. These are nothing important, right, for us. So if you go to chromosome 7, see, this is the one. We know from permutations, it should be greater than 5.7. So on chromosome 7, our QTL peak is between 30 to 36 centimorgans. Even though we don't have a marker there, using likelihood ratios and other things, this is our QTL peak. Let's do the same thing with the other chromosome. So let me start from not the other chromosome, other trait. So for that, let me start again. Open QGene, file, load data. Saved all files. QTL mapping data is the trait I'm interested in. So let me look at the trait analysis. Looks okay, normal t-test, okay, not bad. Let me close this. I don't need any transformation. Analysis, QTL mapping. Yes, I said there are multiple types, but I'm only interested in composite interval mapping for now, most used. 51 is the trait. Select all chromosomes, shift, and everything. Let me first click on log. Yeah, greater than 3 makes sense. Possibilities on chromosome 7 again. Okay. Resampling, sorry, options, select cofactors, do the stepwise factor selection. Okay. Again, a simple pick. Resampling, permutation, make it faster, do 1000, but I'm just doing 50 for now. 3.7, 4.8 are the cutoff. This is 0.05 level and this is 0.01 level. So 3.7 is the cutoff. So options, export text to clipboard. Chromosome 7. So this is 3.7 is the cutoff, right? 30 to 36 centimorgans. Okay, the same thing like before. Next, a few other things that we're interested in. What contribution does it make? So I'm taking an R square on the log, the additive effect. Those are the three important things. So file, export text to clipboard. This is chromosome 7, and the additive effect is, these are the additive effects, and these are R squares, meaning the QTL has approximately 12%. I mean, the variation explained by this QTL is 12% of the phenotypic variation. Okay, so 8.7, 11, 12. So I'm just averaging it out, and these are the additive effects. And if you want to save the figure, there are a few options. And if you want to only do the 7, for example, a horizontal zoom, vertical zoom that you could save directly. And this is the R square right here. If you want only the R square, you can say this 53% explained by the overall thing. The log, the additive effect is plus 79 effect. So you can play a bit of playing around, changing colors and all those things. If you want to have this view, and zooming in here. So this gives you publication ready graphs kind of, or else you have other softwares. So this kind of marks the end of our QTL analysis. So if you have any questions, I'm happy to take on the QTL analysis. So these are the things I explained here. Single marker regression, 5 options to save images and text, composite interval mapping, selecting cofactors, change it to stepwise cofactor selection. You see a narrowed QTL region. You can do multiple traits at once too. In this example, I selected both the traits at once, and you can export the images. These are the cutoffs. For a second, I thought I was not on Zoom. All right, so as far as my experience goes, Carthagene and Qgene are simple click of buttons. All we need to know is like what has to be clicked on. So moving to the third part, a little bit complex, association mapping. So, so far we looked at linkage map and QTL analysis. With the QTL analysis, we're able to identify regions of the genome that are associated with the trait. But one drawback is it is only limited to the cross we are studying. For example, in the US, there's a cross A by B, and we identify a QTL of chromosome 7 for plant height. But in India, we have another chromosome C by B. Not necessarily that it could be on chromosome 7, it could be completely. So that is one drawback. It is population by population, may or may not. One advantage over that is the identified people have a very high variation, meaning it explains 30%, 40%, 15%, a lot of variation explained by QTLs. So to counteract some of the problems with QTL analysis, let's say, like if you take humans, for example, you cannot develop a cross. So how do we identify regions of interest? For that, we started using a method called association mapping that has been extended to plants. The basic idea is you take a set of lines or samples which are diverse enough that show variation in traits. Say, for example, if you take plant height, you have take some varieties from US, some varieties from China, India, worldwide, or in India, where in different regions, cultivars, land races, could be anything. Just take them, phenotype them so that you get a continuous trait variation, genotype them, and try to identify regions of interest based on them. Sorry? Sorry?
Your voice is breaking. Please increase some volume.
Okay.
Increase volume, sir. Voice is breaking. Voice breaking.
Can you hear me now, right? Better?
Little more, can you increase, sir? Volume.
But volume increase doesn't really help, right? I have to go with some mic settings. Okay, let's see. Something. Is it better now? Is it better now?
Yes, Sujan.
Yeah. Okay.
Okay, sir.
So we briefly discussed about the marker-assisted selection, right? So compared to phenotypic screening in different places, few hundreds of samples, will be just doing a few of them, decrease the number of years. So basically, for this marker-assisted selection, so far in the last 40 years, we have been using QTL mapping, and association mapping we have been using in the last 10-15 years. So this I already explained before. So basically, association mapping is also known as linkage disequilibrium mapping. It is used for the same purpose of identifying regions or genes of interest that cause phenotypic variation. So basically, we have to understand the concept of LD because this association mapping is based on this principle. It's basically a non-random association between two markers or two genes or the two QTs. In this example, on the left, you see that locus 1 and locus 2, where whenever it's an A, it's a G in locus 2, and whenever it's a T in locus 1, it's a C in locus 2. But this may not be related. Here, the locus 1 and locus 2 are correlated, or in genetic language, they are in linkage disequilibrium. But if you look at the B, it's the same, but it is associated with a trait. So now you can use these two markers as a proxy for this particular seed code color. There are two measurements that we use for LD prime, which only takes a recombination history into account, and R square, which takes both recombination and mutational history. R square is the most commonly used if the value is between 0 and 1. In simple terms, it's nothing but the correlation squared. So there is another concept called as partial LD, where we take population structure as a covariate, because sometimes population structure and kinship could cause bias in our LD estimates. When you look at association mapping papers, you often see LD decay and LD heat plots. These are nothing but the distance over which LD persists and will determine the number and density of markers that are required. As an example, if you look at this particular, this is for two populations, approximately 130 samples of soybean, about 35,000 things. So here what I'm trying to say, each gray dot represents an LD between two markers. We did a nonlinear regression to get this black line out. So here, what I'm trying to say is, my LD for this population is approximately 2 megabase pairs. So to get a reasonable estimate of for identifying all the genes or all the QTLs in soybean, I need to have a marker every 2 megabase pairs, and soybean is 1000 megabase pairs. So I need, say, 500 minimum markers for identifying the QTLs. So the higher the better it is. So that roughly gives an estimate. I think, yeah, it's the physical distance, but it's in KB. Sorry, it's not the megabase pairs. It's 2 kilobase pairs. We need one marker every 2 kilobase pairs, and soybean is approximately 1000 megabase pairs. So you need a few thousands of markers to get a reasonable estimate. When you have longer distance LD, meaning your LD extends a few centimorgans or few megabase pairs, you can just have a few markers for your study. When you have a short distance LD, meaning it's only at a few base pairs, then you need a lot of markers. So let's suppose you have LD of just a kilobase pairs and you have a 1000 MB, 1 GB genome. So you're talking about a few thousand markers that are necessary. This is one other example. On the x-axis, you have centimarkers. Here we are seeing only have 1500 markers, and here I take generally 0.5. Some people take 0.1, but I'm skeptical on using that. I use 0.5 wherever this reaches, which is approximately 2 centimarkers, is the cutoff I have. So with the distance, LD will reduce, and every generation of random mating, LD becomes 1/2. As the number of generation increases, LD decreases. So let's take a standing population or a natural population, say somewhere in some hilly regions, you have a natural population of a plant. So every time a selfing or crossing, you have over time many generations. So in some of the generations, you have mutations, or if it's a cross-pollinated, some other recombinants or crossovers in them that leads to standing variation. So as long as the number of generations increases, your LD decreases, or the correlation between the markers decreases. Because whenever a mutation or a crossover is introduced, it's not introduced in your entire population. It's only introduced in only a few samples of that entire population, right? So when you take land risks and wild types, they have been hundreds or thousands of years, so they have limited LD, or your LD is only a few kilobase pairs. Whereas if you take cultivars, which have been developed in the last 70 or even 100 years, it have long LD because it's just a few generations. LD heat maps is this one. So basically, it's just visualizing the R square values. So for example, I have this GM763973119 that's highly significant. If I look at here, there are two genes close by of FRO2 and FRD4. So I can look how are they related. So if you look here, this particular marker, even though it's not that good here, red, this thing, if it is light blue, they're in good LD. If it is red, they're good in LD. No, the other way around. The red and oranges are the high LD regions, and the blue are the low LD regions. So you can see these are related by orange, meaning these markers could be a proxy for this particular gene for association mapping. And basically, we need to understand linkage disequilibrium for starting an association mapping experiment. There are various factors that affect LD. One is mating system. So if it is selfing, reduces the opportunities for effective recombination, so there is higher LD. Whereas if it's a cross-pollinated, you have crossovers, higher recombination, so your LD distance decreases. Domestication. So in domestication, what happens is after a bottleneck, some haplotypes will be lost. In this particular case, you see 4 or 5 haplotypes in the ancestral state, but because of domestication, you had only 2 of them selected. So this results in increased LD. And some other things that cause increased LD include genetic drift, gene flow, positive selection, population structure. Let's not go into the details of all these things. And there are a few other factors that decrease LD. One is recombination, because LD is broken by recombination because you have crossovers. And a population, the larger the genetic variation, the faster is the LD decay. For example, LD decays within 1 kilobase per in land races, 2 kilobase per diverse inbred lines. And you have mutations and gene conversions that could reduce the LD. So should we have a long increase or a decreased level of LD? Meaning for my association mapping, should I use the cultivars or the wild types? Well, both of them. If it is a long range of LD, meaning if you use cultivars, you just need excuse me, coarse mapping and fewer markers. But if you are using wild types, land races, there is short LD decay, you need 5 mapping or more markers. And there's a higher chance of identifying more cuticles too, because they're diverse too. So with association mapping, you have 2 kinds: genome-wide association mapping and candidate gene. Candidate gene is wherever you think a particular region is significant, and you just extract markers from that region and do association mapping. But most of the time we use genome-wide association mapping. So one advantage of this association mapping is higher resolution. It's just a few base pairs of resolution, meaning from the gene of interest or from the variant of interest, your identified markers could just be some 100 base pairs or a few kilo base pairs. Whereas if you look at say recombinants or near isogenic lines or everything, it is approximately 1 MB or 10 MB. So this is one classic example that shows the resolution differences between them. In dihybrid, you see it's 10 centimorgans, and rails you can get until 5 centimorgans, whereas if you take a diverse germplasm and do GWAS, you get less than 1 centimorgan. So what exactly is association mapping? So let's assume you have a low LD-SNP on your left right here and a high LD-SNP on your right here. And this is one phenotypic data, well remember. And this is not the sample. I mean, this SNP or this necessary variant was not sequenced. So we don't know this. So if you look here, the correlation between this and this is low, and the correlation between this high LD-SNP is high. So based on this particular numbers, if you look at the means, mean of G and mean of A, the P, you see that it's on 0.77, meaning it's not significant. Whereas if you look at this 0.037, meaning this marker is close to the gene of interest. But we did not sequence this, but in case we sequence this, it's still a lower value. So association mapping is basically more like a t-test in case you have 2 classes, ANOVA in case you have 3 or more classes. You just compare the means for each marker for the traits. And there are two, before I go there, let me quickly look at the chat. Okay, the PPT is already in the GitHub. So problems of association mapping, if you have a population structure, let's assume, meaning population structure in the sense, let's take say rice varieties grown in Andhra Pradesh and rice varieties grown in Tamil Nadu. For sure, they have different genetic backgrounds, so they could belong to 2 subpopulations. So due to this soil and climate differences, let's suppose we have a T in a particular location in Andhra cultivars and a G in the same location in the Tamil Nadu cultivars. And let's suppose when you take say number of pillars, there could be some minor variation between Tamil Nadu and Andhra. In that case, you can associate these TG differences as a significant marker, but that is not, it's a false positive. That difference is mainly due to population structure because the cultivars from there form a subpopulation, the cultivars from Andhra form a different subpopulation. So as an example, I had high disease resistance for 2 subpopulations and an SNP. If you look at this SNP1, by quickly looking at it, you can say this SNP is related to plant height or plant disease, but that is not true. That could be a false positive. So for this, when using an ANOVA or an equivalent F-test or a regression, we need to use covariates that control for population structure. And the other one is kinship. Kinship is family relatedness. Say, for example, if you take cultivars, their parentage, if you go back, say 5 or 6 generations or even more, you could see they're mostly related or have similar parentage. So we need to control for that relatedness too. So the main difference is QTL mapping. We use biparental population, whereas in association we use natural population, and we have limited number of recombination events there and many recombination events here. Low resolution, a few centimorgans, whereas a few base pairs here. This is QTL mapping is good for discovering rare alleles, whereas association mapping is only good for common alleles. Another comparison is let's not go into the details. A few more comparisons on what LD and linkage are. Since our time is limited, I'm just directly going to the methodology. Sorry about that. So for the GWAS methodology, there are a few steps. One is we need a population, a diverse population and phenotyping of it. Then we need marker information. Then we need imputation and minor allele frequency filtering, a few covariates, a regression, model testing, and QTL and significant markers, and finally validation. So population is, let's take a population with overall diversity. For example, diversity land throughout the world. Or you can take, say, cultivars used in a particular state, say all the rice varieties grown throughout the world or whatnot. And then phenotyping, it's better to have data from multiple locations so that you can capture the effect of climate, soil, and other things into it. So you can analyze based on individual locations or combined locations. A question that comes is, say, for example, I have a field trial in Tirupati, field trial in Vijayawada, and field trial in Guntur Lampham, for example. So should I analyze it separately or should I analyze it same? So the answer to that is, if you intend to develop a variety for Andhra Pradesh as a whole, do a combined analysis. But if you intend to develop a cultivar only for Andhra, Guntur, Vijayawada region separately and Tirupati region separately, do the analysis independently. So when we even want to do combined locations or combined replicates, use an adjusted entry means where it considers the effects of for replicates or locations, all those things as random factors, and nullifies some of those effects. So as an example, if adjusted entry means is I had 143 genotypes from 3 locations, 4 reps and 2 ratings. So considering G by E interaction, if I look at the G adjusted entry means they capture some of those effects. Say for example, this is the difference that can cause a serious effect downstream analysis. So the second step is, so I have a population now, I have some marker, I need some marker, and I have some phenotyping for the trait of interest. The second step is I need to have some markers. So it could be SSRs or golden gate assays or high density using affymetric shapes or GBS or NGS data, so on. And the third step is imputation. With QTL, if you remember, we used markers even with missing data. But with association mapping, you cannot have markers with missing data. So there's a procedure called as imputation. So the idea is we predict the missing phenotypes based on likelihood. So if you look at the figures in the bottom, when you do association mapping with missing data, only a few markers have been used for association analysis, whereas on the right, since imputation has been performed, you have more markers, you have a better resolution. There are many softwares. I use Beagle. We have version 5, which is good enough. So basically the idea behind this is, for example, you have a raw data, question mark, missing data. So it makes haplotypes, meaning 1 and 3 are really close enough, and 2, 4, 5 are close. So it assumes since these are close enough, this could be a T, this could be a G, estimates likelihood, or this could be a T, or this could be a C. It takes multiple possibilities, probabilities. Let's not do the statistics behind it, and finally gives you some estimated values there. Okay, now we have our genotype. Say we have some 100,000 markers. We imputed them. Then do we really need all the 10,000 markers? As mentioned earlier, GWAS only needs markers that are above. It's not useful for array release, meaning out of a population size of 1,000, if a variant is present in only one sample, your ANOVA or your association mapping will not show that to be significant. So those are the markers we have to remove. So for that, we use a minor allele frequency of 0.01 or 0.05 and remove the markers that have very low minor allele frequency. Then the next thing is estimating the covariates for population structure and kinship. So structure is one software, PC is the common software that we use for populations control. We'll talk about all these things. As I mentioned earlier, association mapping has a lot of new concepts. So if you did not really work before, might look complex, but on a re-revision or a rereading, it makes a lot easier. It's really good and easy. So we need population control, as I said, to control for any false positives. This is one example in humans where we had population 1 where blue alleles are more and population 2 where red alleles are more. At the same time, you have cases on the cases and controls that will be different. So you can say that this marker is related, but indeed it is not. So this is a population structure example where we had like 5, 6 subpopulations, each one is a different color belonging to different regions. So we need to control for that as a confounding factor or a cofactor to avoid those false positives. So to avoid this, there are multiple options. Structure is one, but it takes time, so let's not do that. The other one is the principal component analysis. Basically, it infers continuous access of genetic variation. Your 1,000 marker data, for example, is condensed into some 20 columns are 20 principal components that captures all the information. So we could directly use that. So a PCA plot with something, this is in beam. A PCA plot with a few markers shows something like this. So it's able to distinguish between the two kinds of common beans, Mesoamericans and Andeans, and cultivars and races within. But the question comes is, how many principal components should I take? Well, you can take the top 5, top 10, something like that, or the amount of variation explains it, 25%, 50% variation, so on, or some statistics like some maps or trace evidence distributions, something like that. Or you can use stepwise regression to identify them, PC1, PC3, PC7, so on. Well, in our case today, we'll be using only 25% variation. The top, as you know, principal component 1 explains the most of the variation and it keeps on decreasing. So the cumulative variation explained, if it is 25%, we'll be using that to control. And kinship is a random effect. It is used to calculate genetic variance and the residual variance. This is generally refers to recent common ancestry. As I said, if it has similar pedigrees. And here you have identity by descent and identity by state. Let's not worry about this. And the next step is regression. Here you have two main models. One is GLM when you have only fixed effects, and MLM when you have random effects, meaning when you have kinship as a random. So regression analysis is performed for one marker at a time. And we have genetic variance and residual variance estimated for each and every marker. We call them exactness. It can be implemented in SAS, MA, FastLM, many softwares. But to maintain, to decrease the computational burden, about 7-8 years back, we had approximate tests. So where the genetic variation was estimated only once and used, that saves a lot of time, but not anymore. Now we have really fast software. So other than the models ELM, MLM, it could be a Wilcoxon test. If it is a non-parametric distribution, logistic regression, generalized linear model, if the phenotype is discrete and so on. And there's a software called as GAPIT. It's an R-based, it's all command-based. It's a very good software, just like Tassel. Excuse me. That runs additional models other than GLM and MLM, which are MLM super performs if you blink so on. So which marker to run? So the idea is like we run multiple models with and without kinship, with and without population structure, and we take the best among them. So for example, today we'll be running about only phenotype is directly related to marker, assuming there is no population structure and no kinship. Then with one with structure, one with kinship, one with structure and kinship. So the idea is the best model is the one where the residual errors follow a uniform distribution. For that, there are softwares, but I developed a method called as MST based on observed p-value and expected p-value. QQ plot is a visual representation, so not necessarily we'll be able to get the exact information. So for MST calculation, I included all the steps and I'll talk about in the end. So once we estimate, say out of the 4 models, I got say model B as the best model. What should I do next? Well, you can sort by based on p-value, based on a one for any correction. Let's assume we have 10,000 markers. So you divide 0.05 divided by 10,000. So that is 5 into 10 power minus 7 is your cutoff. If a marker has a p-value less than that, that is significant. Or else you have FTR, PFTR, which corrects for multiple family corrections. At the same time, you have bootstrapping, simple regressions, and many more. In my case, I use a different kind of bootstrapping. If you read, I had around 15 to 18 papers on association mapping. So if you read them, I used different methods for different species. So, and I wrote why we had to use that. You can get that information there. Well, let's suppose out of those 10,000 markers, I found some 25 markers that are highly significant. And I know the location, I mean the chromosome and the positions. How are they useful? If you look at a particular location, there could be some 5 or 6 markers side by side, but not all represent a QTL. It is just one of them that represents the QTL peak. The others are simply in linkage disequilibrium with this QTL peak. So to address that, I proposed a method called a stepwise regression, which is a statistical methodology. So it takes all the 6 markers into consideration and says, hey, this is the important one, not the other 5. But if you look at, let's assume you have 3 QTLs and you have 15 markers overall. So if you include all the 15 markers in stepwise regression, you end up having only 3 markers in the final regression, saying that there are 3 QTLs. And all other last markers are significant, but they are in LD with the QTL regions, excuse me. And you can look for epistasis. Let's not go into details of this. And how much variation does a marker explain? For example, if you look at, remember QTL mapping, the variation explained was 10%, 15%, even 53% in our case. But here it's a bit tricky. We estimate the variation explained to be 1%, 2%, because here the recombination is limited to only a few number of samples, right? And there are multiple methods. So in our case, let's just look at the tabular output to get the variation explained. And what's the effect of allele? So let's say we have a reference allele of T and the mean is 10, and we have variant allele where the mean is 16. Say plant height, for example, the additive effect is 3. So 1/2 of the difference. Meaning from T to G, if there is any change in allele, it increases the plant height by say 3 centimeters or 3 meters, whatever your unit is. Then we do some marker combinations. So the idea is to get which marker, no single marker explains the entire QTL effect. So it's some the combination of multiple markers or multiple QTLs that causes the trait variation. So we need to identify that combination. So let's assume you have 3 markers, all are biallelic, right? So you have 8 possible combinations. So for an SNP, for example, this is the IDC score. 5 means it's highly susceptible and 1 is highly resistant. So I have some a testing sample of 1,000 samples. I take these 3 markers, test them. I only take if it is a BBA or AA or AA because they are less than 3. Or the other ways, I discard ABB, AA, and triple B because they're greater than 4. So that reduces the number of samples I could use further, which is marker assisted selection again. The functional aspect, okay, this marker is significant. How is it related? If you have the reference genome and the annotation for that particular genome, you can say, well, this is a synonymous substitution, this is in the gene, this is in the promoter region, or this causes this effect, or whatnot, all those things. So basically, we had a phenotype from multiple locations or multiple tribes. We had the means or the adjusted means, and then we had markers where we did the imputation, minor allele frequency, estimated PC and Q shape. Then we do regression, model selection, identify significant markers and do stepwise regression, and finally get the allelic effects and do some SNP annotation. Okay, we identified markers that are significant. What should be done next? One thing is we can evaluate them on different genetic backgrounds. Then we can verify them by using, so say CRISPR is the recent one, or else you have WIGS and many other knockdown silencing experiments. And we can use directly for marker selection in the breeding process. One problem with association mapping papers is they are not being validated. They just publish a paper, hey, these 10 markers are significant. So breeders are not really using them. So, but if you could validate in say 2 or 3 additional populations, say with the breeders, all the population he has or all the cultivars in that region, that way once we validate, it's more confident. One example I want to point out is I did a cadmium evaluation in durum wheat, which is used for pasta and noodles preparation. So for each phenotyping, it takes $38. So for developing every time they had a few thousand samples, so it's too costly. Using association mapping, we developed 2 markers and we developed 4 allelic combinations. So out of which 2 are good or resistant or have a very low cadmium content. And our marker system for the 4 markers costs us approximately 50 cents. So $38 versus 50 cents for 10,000 samples every year. That's the advantage if we could identify the markers. This is the one I was talking about, cadmium accumulation in wheat. I want to quickly point out, as I said, I have many papers. I want to quickly point out the type of population and the type of markers. here these are near inbred lines, and this is from using a bead chip of 9,000 markers. And here is in barley, it's 8 breeding programs from 4 years, 96 lines each, and we use 4 regression lines, and we got a QTL peak at combined method, all the 32 things at chromosome 5H. But if I do the same thing independently, where the location is A, B, B, A, MT, and NT, see the same trait. You see the variation in the number of QTLs and the effect of QTLs based on the log P values. And in lupus, for example, I used plant introduction, say FLPs. In Fusarium head blight, I had background BC1F7s and BC1F6 that I used, and I used dark markers. For iron deficiency chlorosis, I used advanced breeding lines and golden gate assay, and so on and so forth. So the idea is we can use multiple kinds of populations as long as they're diverse for the right of interest, and you can use different marker systems based on the availability. So that completes my theoretical part. Any questions so far?
Sir, for GWAS, how can we perform in Linux system or in Windows?
In Windows,
Can-
as it does it, I'll show you that.
So one practical, sir, one sample, can you show the GLM and MLM?
Yeah, I'll show you that.
And Manhattan plot and Q2 plot also, please.
For candid gene-based association mapping, what number of markers will give good results? Well, based on the LD or population, I cannot say, for example, if it's highly heterozygous, like say switchgrass and a polyploid versus common bean, which is diploid selfing, the number of markers varies. Depends on your crop system. But if you have, say, for example, one for every 10 base pairs, it's very, very good, but I don't think that's feasible. What should be the ideal size of LD mapping population? Well, minimum 100, 150 is the one. Minimum, I would say 200 ideally. And these are some good references if you want to have a good understanding of LD, concepts of LD, what affects LD and all those things.
Sir, my request is at least can you do one sample in GWAS in Linux system for practical through command based.
Well, we need those installations, and the problem with the Linux based and command based is, you have to. Not many people are aware of the command system, so it's so hard, and you end up with a lot of questions.
Yes, sir, because that is important, sir. Actually, I joined for this one only.
But there are two softwares which are in R. I prefer those. One is Gapit. So it's in R. If you're into command based, it can work on Linux, Windows, anything. This is a really good one, but it's all command based. But you can write a script, R script, and feed it into it. So it runs all of them at once. All the functions are very well written. Just the other one. It's one other software. Tassel has an R function too, in case if you're interested, that works on Unix too. But if your Unix is GUI based, you can use Tassel actually. So let me.
Because actually I'm expecting some practical set.
Well, I had to keep all the 250 people participants in mind, so it's a bit hard. But
Actually, what are the steps to we need
so,
to
but
follow
if you Linux, are
that,
you using a terminal window or using a GUI?
yes,
What are you using? Command line? I mean, terminal?
yes, sir. But
Yeah,
right now, we can also do yourself, sir. We can practice later by using your video.
So basically, it's like file open as, okay, I'll walk you through all these steps. I wrote everything in detail, what has to be done. So let me open as this is jar file. So if you're using a Linux with a front end, GUI end, in that case you could run there. So the first and the foremost step, file open as, I use sort positions, make best case, or else you can select the file you want. Okay. And here I have my genotype, my phenotype. Those are the two things I need in general. For formats and everything, look at the TASL manual. So this is my two traits. This is 1 and this is 2, and this is my genotype data. As I mentioned, we can impute it here, but in my but these are different algorithms. I use Beagle for imputation and then convert into this format. But these are rather the imputation methodologies. Now let's filter by filter sites, minor minimum frequency management. Okay. Remove minor SNP state. Fine. So minimum count is 5 out of 136. So that counts to, or maybe let's keep 7. That counts to approximately 0.05% minor allele frequency. No, nothing has been removed, meaning everything is within the minor allele frequency, because the file I created has been edited to account for all those things. So as I mentioned, the first step is phenotype, genotype, then you have the filter by minor allele frequency, then estimating the covariates. For that, I use analysis, relatedness, PCA. Let me do first to say 10 covariates, sorry, 10 components, return eigenvalues. I don't need eigenvectors. Okay. So these are the PC1 to PC10. If you look at the eigenvalues, look at the cumulative. The first one explains 10, second cumulative 15. So 4 principal component explains about 25% of the cumulative variation. So that will be my covariate for regression. So I'll remove this file, delete dataset, PC, file, delete dataset. I will do for analysis, relatedness, PCA. I know we need only 4, so I'm selecting 4. Don't need eigenvalues. Okay. Estimated principal component analysis that controls the population structure. The second one is relatedness, kinship. Okay. I loaded the recent version of TASL and there was a problem with a plugin something. So it's not able to estimate kinship, but the previous versions of TASL does. So I suggest, in case if you're using the latest version, write to the authors asking, showing this issue, they'll immediately fix that. I will do that too, but I urge you to do that too. So since we cannot do it right now, I have another option. My another laptop, my work laptop, I was able to. So I am uploading it directly. This is the one it is. There is a kinship one. So basically it has 136 by 136 matrix. The relation between these two. But in case you want to do analysis, relatedness, just do a kinship and it select the defaults and should automatically do that. So we have the phenotype data, genotype data, genotype data that was filtered. We had the pieces, we had the kinship. So we got all the requirements. Just a quick follow-up. So we got a population, we got a phenotyping. I already gave you the adjusted entry means, and the genotyping marker data, I already imputed it. We did the minor allele frequency, nothing lost. With the population structure and kinship to control for population structure and kinship. Now let's come to the regression. So first thing is genotype and phenotype and the phenotype, select both of them, data, intersect join. This has only phenotype and only marker data. So now let me rename this as name for my understanding. Second, this is phenotype, genotype, and let's include the pieces. Data, intersect join, intersect join. I had this which has pieces, phenotype, and the genotype. Let me rename this as PC. So I made two new data sets that have this information. For GLM, I don't have any random effect. So naive doesn't have any random effect or it doesn't have a kinship. So naive, select on that file, analysis, association, GL. So let's say save to file. I said naive stats, then this one. Naive effects. We don't need anything of this. Biallelic only. Output site stats. So I'm just saving everything. That's it. Okay. So I'm done. Let's check here. We have all the naive effects and the naive stats of them, non-empty files. So now let's do PC4 analysis, GLM. Start. Done. So we're done with the naive model, which has no structure, no kinship, and we did one model which has only structure. Now let's do the next step of only kinship. So for that, I'm selecting this file and a kinship. Hold down the control key and select the kinship file, analysis, association, return MLM. Okay. Basically, the default, as I mentioned earlier, for the various components, you can do only once, but I prefer using this and using no compression. But it can take up to an hour or so. So to avoid that, I'm using this for now. But in theory, I prefer this because of some of the statistical issues I saw. So this is the one I prefer. But for just for demo purposes, I'm using this because that's less than less than couple of minutes. Let's hit run. Write output to file, browse. I don't want to filter. Okay, it's running fast. But for example, if it's taking a couple of hours or more than that, you can simultaneously start 4 or 5 analysis at the same time too, as long as your system has a memory. Now let's do these two together. So PC4 matrix has phenotype, your principal components that control for population structure, and your kinship. This is the fourth best model we're using: analysis, association, MLM. We're using MLM because kinship is a random variable. And again, this is the best one, but for now, this one. So we're done with all the things. We're done with both GLM and MLM. So, so we did phenotype is marker, phenotype is equal to marker plus structure, marker plus kinship, marker plus structure plus kinship. So we did all these three. Then the next step is model selection. Oh, before I go there, let me show you the output files, what they look like. So let's start with naive model. So with naive effects, these are the estimated effects for. So with the change in LD, this is what happened, minus or plus some of certain values. These are the ones we're interested in. See, here I started with two traits, right? This is one and this is two. So both of them are present. Your marker name, your chromosome. I mean, I didn't give all these things. The chromosome position, these are, and this is the one, the p value which we're interested in. And this is the R square which we're interested in. So let me quickly show you. So let me sort the data. Sort first by trait, then by marker p value. If you look here, say let's assume minus is our cutoff. These are the significant markers. But if we know the chromosomal position, that will be really helpful where the QTLs are. Okay. So similarly for disease 2, we find that. And this is the marker R square mean. This one explains in 200, 12% of phenotypic variation. This one explains 10, this one explains 10, and so on. And as you know, if you sum them up, it is greater than 100. When you combine multiple markers, the effect is kind of a confounded by few, so the total sum is different from the total variation explained. And this is the marker. These are all unnecessary columns which you don't read. So these are the two columns, p-value and the marker R-square are the ones which we look at. So for model selection, okay, this is name, right? Now for PC2, we get the same things. Now let's looking at the kinship. Let's not look at the effects. Okay, here we have trait marker, chromosome position, the p-value, the additive effect, dominant effect, marker aspect. This is the one we're interested in. So this will the additive and the dominant effects are mostly NANs based on the information we give. So it's marker R square and the p-value that we are interested in. So out of these four models, what should we take? So for that, come on. Let me take name statistics, control A, control C. So we're interested in trait, disease, and p-value. I'm removing the unnecessary ones. This is the stats. See. So on, we need to just keep those four columns and remove them. So the way I did was for mark this one. For each trait, for each model, sort by p-values, assign a rank. Okay. So let's remove this list two just to avoid confusion. I'm just working with the two models at now. One is a name model and this is a PC model. So for that, the steps are for calculating MST for each trait and each model. Data sort, custom sort by p-value, rank. This one, two, three, so on. So I got the rank because they're sorted p-value. And the next step I need is for each thing sort p-values, assign a rank 1 to n, where n is the number of markers. Expected p-values divide rank by n. This one will be 1 divided by 6940. So this is our expected p-value. Okay. One.
So it's E2.
Sorry.
It's E2. E2.
Oh yeah. Thank you. Thank you for it. This is our expected p-value. Then we have expected p-value minus observed p-value and square the difference and mean it. Difference. This is the MST value. As you are aware, MST refers to mean square difference. So we calculate the difference, then we squared it and we did the mean. So this is this much for this particular model. Similarly, we need to do it for the other ones. So basically sort, custom sort, p-value, okay. It automatically gets updated. So this is 004 and this is 0003. So if you remember, this is the new model where we did not control for population structure. Whereas this one, we had a 0003 where we controlled for population structure. So comparatively, controlling for population structure is a better model than the regular model. To confirm that, to see if there is a population structure or not, Dazzle has an option called as go to results, go with results, go with LD plots. Results go with PC1. So if you could see these samples, all the samples are scattered. This could be one population, this could be one, and the middle one could be one. So basically, there could be some 2 or 3 based on statistics. Different statistics, you might end up with some 2 to 5 subpopulations. Let's for suppose if you don't have any population structure, then you'll see it a single large cluster where you cannot distinguish between the groups. But here I could kind of distinguish between the groups visually. So, there is population structure. Since we control the population structure using a principal component analysis, principal components into our regression model, we found that to be a better thing. Similarly, we can do it for kinship and PC plus kinship. So out of the 4, we take the best one. For now, let's suppose this is the best model. So these, if you take a bond for example, it is 0.05 divided by 6940. So if your marker is less than 7.2 into 10 power minus 6, it is significant. In our case, none of them is significant. So we can do FTRs, PFTRs, or let's suppose minus 3, 10 power minus 3 is the cutoff. So these are the 3 significant markers we have for this particular trait. Similarly, we can have other traits as well. So, and R square explained by this is approximately 8%, 7.9%. As you could see, the p-value is the same and R square is the same. So these 3 markers, I believe they are from the same chromosome. In the data provided to you, I did not provide you the chromosome and the positions, but basically this data is from Harley, 1H to 7H. So I just removed all of that information as much as I can, and has been manipulated a bit. So that because we don't want anyone to take this data, publish or do some other things. So that gives you the basic idea of significant regions or the significant QTLs in an association mapping. Let me repeat the TASL main steps again. I mean, there are other steps, downstream interpretations. I'll not be talking about those given our limited time. So the first step is open TASL file, open as, make best guess, sort position, select all the files you want. In our case, I'm just selecting for now the phenotype and the genotype, 2 files, hit OK. You get all of those information. The next step is if you want to filter based on missing data, minor allele frequency, use filter sites. It creates another subset if there are any. If not, it will stay the same. Then hit we need cofactors, analysis, relatedness, PCA. First, let's go with say 10. Make sure you get the eigenvalues. Okay. From the eigenvalues, see how many you want. Maybe if you want say 50%, you need 15 of them. So let's take 41. Let's take 25, which is 4 of them. So remove this thing. Remove these 2. Do again file analysis, diversity, relatedness, PCA. 4. Don't need eigenvalues now. Next, basically analysis, relatedness, kinship should give you a kinship matrix. Unfortunately, it's not today with this newer version of TASL. So I'll just upload the ones we had already. So that would be that. Now we need to run regressions. So for that, the first step is genotype and phenotype has to be merged together. Select both of them. Data intersect join. You can leave it this way if you want to. Oh, sorry, this is the PC. Wrong. Phenotype, data, intersect join. You see genotype and receptor. So this is our name model, this is our PC model. So name model analysis, association GLM, save the files. It's okay. Here it else it's giving, so not worry about it. Then for the PCA analysis, association GLM. Okay, it gives all the information here. Then we have kinship. If you have kinship, select that particular file, kinship together, and hit analysis, association, MLM. No compression, re-estimate for each marker, but for now I'll be using this. Run, right out. Okay. Done. Next, this one and this one, analysis, association, MLM. So save all the output files, copy these output files into Excel, just including p-value, calculate the MST, select the MST and take the highly significant markers because they're sorted based on some PFTR, FTR, or some permutation tests, or just a random cutoff based on the trait and the population under study. So this completes this association. In addition, we have other options called as we can generate an LD plot, we can generate a QQ plot, for example. Let's suppose this one, analysis, results, QQ plot. We just need this is one. This is the QQ plot. And for Manhattan analysis, results, Manhattan plot, select it, this is one. Because we don't have any chromosome positions, it's assuming everything to be in the same chromosome. Okay. Chart is if you want to look for histograms or XY bars chart, any of that information. Optimal number of pieces, I generally use 25% as the thing, but it depends. There are multiple methods of selecting the number of pieces. Welch score, MAP score, 25%, 50%, top 5, top 3, or some other criteria. I mean, like once you start working with your data, based on your data, will try to decide, or else based on some previous literature, you will be able to decide. So LD plot is calculating it, takes bit of time. Invalid selection LD. So we need to diversity, linkage, equilibrium. Okay, I'm estimating LD basically, but it's not able to run. So basically LD decay plot, we have two types of plots, LD heat maps and LD decay plot. The one you are asking is the LD decay plot, which is nothing but this. LD decay plot is, it shows approximately genome wide on an average how long does the LD extend. This is useful to justify the number of markers we are using for our analysis. Let's suppose we use 100 markers only for a 1000 megabase per genome. In that case, we have one marker every few megabase pairs. If your LD is only 100 kilobase pairs, meaning you are saying your work is crap. But if your LD is say 1 megabase pair and you have some 10,000 markers spanning one for every kilobase pair, you are good to go. So for that, we use a non-linear regression. So you calculate an LD A by B, A by C, A by D, so on for each and every chromosome independently, combine all of them, do your non-linear regression, and you get a black line like this. So wherever it catches 0.5, that is the spot until where the LD extends. 0.5 LD is nothing but 0.7 correlation, because LDR square is nothing but correlation squared. Since 0.7 correlation is a significant number, I use 0.5. LDR square is the cutoff. Yes, the advantage of Tassel, Gapit, or all these things is even if you have 200 traits, it can do all of them at once. You don't need to do one by one. That's the beauty of this. Sure, I'll share the video once this is done. I'll upload it. I'll send it to sir. I don't know where sir is sharing it. I'll share the video with them. But as far as the all the data used, all the PDFs we uploaded into my GitHub site. In the future, if you have a question, I have a Google Forms that will be active for the next couple of days. The next couple of days, Google Forms will be active. You can submit your questions there. But in the future, if you still have some questions or some information, you can catch me up in your LinkedIn. I'm available in LinkedIn as Sujan Mamidi. You can catch me up there and just share your question. I'll be more than happy to address if you have any. Anything else? Okay, thank you. Okay, sir. Tomorrow we
Sujan.
have another session on cluster W the same time, and the day after that would be a holiday.
Done, Susan.
Yes, sir.
So any questions, participants? You can use that questionnaire, which we have already provided in the very first mail, so that the concerned speaker can able to answer your questions. Okay. So I thank you very much, Dr. Sujan, for your nice presentation as well as the demonstration. I hope the participants might have benefited your experience and expertise. So actually, we are trying to upload into a YouTube channel. So I hope today's video also we will upload into the YouTube. So we will send all the links to the participants, maybe tomorrow or day after tomorrow, because just I am testing in the YouTube whether how it works and what kind of things we have to share and all those things. And regarding the PPTs, if the respective speakers, if they are happy to share, then we will share to you. If they have any reservations, that I can't do anything. Okay. Anyway, video we are sharing. Okay, don't worry about those things. Okay. So with these few words, today's session I'm ending. So again, we will meet tomorrow at the afternoon 2 o'clock. Okay. Thank you very much.
Thank you, sir.
Thank you, sir.
Thank you, sir.